Your review slide should look like this. Chat. All right. We're just going to kind of jump right in. So the first activity that I thought would be fun is to take a blank chart and see if we can fill it in. And we're actually going to try to be a little more detailed than the completed figure from like your textbook on this. So this is a pathway that shows our metabolism. It doesn't show the electron transport chain oxidative phosphorylation, but it starts with glucose. And we're going to see if we can fill in all the intermediates and all the pathways. I really tried to cover up glycogenesis and glycogenolysis, but I didn't have the patience to do it on PowerPoint. So those are there. But everything else pretty much, except for a few words, is gone. So I want you guys to help direct me. I will give you this first one. If we take glucose and try not to look at your chapter 22 slides, try to do this from what you know. Try not to cheat. No cheat sheets. So what would be the first intermediate after we take glucose? Before we have really committed to glycolysis, it seems like there's a space right here. Does anybody know what intermediate glucose is turned into? First step.
Is it glycogen? I might be wrong.
Glycogen is not far away, but we're not at glycogen yet. Glycogen, remember, is the long polymer of glucose that's stored in the liver.
Is it the glucose 6-phosphate?
It is. Look at Maggie. Glucose 6.
See, okay. Photographic. Yeah. I can do the pictures.
Okay, Maggie wants all the blank charts. Glucose 6. I'm just going to put a P for phosphate because I ran out of room. Glucose 6-phosphate. Okay. Now, I want to talk about glucose 6-phosphate for a second. This is a very special molecule because there's so many different routes. There might have been a homework question alluding to this. There's so many different directions this molecule can go. We're not necessarily committed to glycolysis at this point yet. There's other things that can still happen. So one thing that can happen is the pentose phosphate pathway. And does anybody remember, this one's kind of the most elusive one. We actually didn't talk about this beyond just looking at it on this chart. Does anybody remember what you make from the pentose phosphate pathway?
I know, like, isn't it two things and one of them has like ribosomes in it?
Yeah, so well, one is for like ribonucleic acid. So if you, I mean, the word pentose should give it away. We're going to make 5 carbon sugars. And these are used for like RNA, DNA synthesis. Okay, but we're also, the one pivotal thing here that is really good to remember, we can also produce NADPH from this pathway. Okay, so this would be the first thing and this would be the second thing. Now, what is NADPH? It's a cofactor or coenzyme?
Coenzyme.
Thank you, Cameron. And it's used to do what? Oxidize or reduce things?
Doesn't it oxidize things, but it's a reduce, like
it's reduced, but it's an oxidizing agent towards other molecules. Yep. Okay, so we've got to have a steady supply of oxidizing agents in the body because as we've looked into many of these pathways, a lot of them are just redox reactions, the electron transport chain and others, okay? So that's the left side. Let's go to the right side. So if we follow the blue, remember the blue, the kind of the color code here, blue is anabolism. Okay, so the building up into bigger molecules. The brown is breaking down, catabolic processes. Okay, so if we run glycogenesis, what's the end product? If we take glucose 6-phosphate and run glycogenesis, what's our end product? Claire, now's your moment.
It's glycogen.
There we go. All right, glycogen. And we store glycogen in the liver. Okay, but then we also see that glycogen can be broken back down into glucose 6-phosphate. Now I have a question on a slide later on where we're really going to analyze these because we did walk through the enzymes that are used for this process. It's really only a 2 or 3 step process depending on if you're in the liver or skeletal muscle tissue, like muscle tissue. But the enzymes are the same except for one. So okay, now let's say we have glucose 6-phosphate. What is the what's another route glucose 6-phosphate could go that we haven't got into yet? The main one.
Is it glycolysis?
Yeah, okay. And glycolysis is a catabolic process, so that's the brown arrow pointing down. All right. We're gonna dive into glycolysis in I think like the next slide. So all I'm gonna do is label that. What is the end product of glycolysis though? We need to put that down here at the bottom of the arrow. End product of glycolysis, we get 2 molecules of
Pyruvate.
pyruvate. Okay, we'll get 2 of them. So remember steps 1 through 5, I will write this in. These are just done once. These are the energy investing steps, but steps 6 through 10, I'm going to draw 2 arrows because those are steps 6 through 10 are done twice. And that's why we get 2 pyruvate in the end. So anybody remember the middle intermediate of glycolysis? Like the one that we get 2, it's the first molecule that we get 2 of, and then step 6 through 10 are run from that. So after step 5, we have what molecule? Anybody remember? Glyceraldehyde 3-phosphate. This is kind of the middle intermediate. So we get 2 of those, and then step 6 through 10 is run twice for both molecules of glyceraldehyde 3-phosphate. Okay. Again, we'll jump into glycolysis a little more in just a minute. Now, pyruvate is also a unique molecule because it can do several different things. It's not just consigned to one fate, and it depends on what the conditions are. So let's look at what happens. Well, let's do the first major product of pyruvate. If we're just going to continue down our pathway for the major metabolic pathway in our body, what is pyruvate turned into?
Acetyl-CoA.
Acetyl-CoA.
Isn't this the step that depending on what happens, it can either be fermented into like an alcohol or a vinegar?
Yeah. So there's fermentation. That's anaerobic microorganisms, yeast. That's the process that I mentioned happens commercially when they make beer. Is fermentation. It's turned into an alcohol. Okay. But in mammals, we also have anaerobic conditions. We just don't make ethanol from it. So pyruvate under anaerobic conditions. So like what parts of our bodies would run this pyruvate under anaerobic conditions would run it to this next product? Anybody remember what particular cells or tissues we mentioned? There were 2 things.
Was it the making of proteins?
Not quite, Madison. So in anaerobic conditions, so it would be like our red blood cells. They don't have mitochondria. So red blood cells cannot, I mean, they're cells just like any other cells, but they don't have the energy powerhouse, the mitochondria, to create ATP. So they have to still obtain ATP to run cellular functions. So they do it through this mechanism right here. They turn pyruvate into, what's the end product of, if we're gonna oxidize pyruvate.
It becomes lactate.
Sorry, we actually reduce it here. We oxidize it to acetyl-CoA. It becomes lactate. So red blood cells and then muscle tissue. Cells in our muscle tissue, they have very, very low oxygen supply. So they will take pyruvate. And remember, y'all probably have heard, especially like you, if anybody's had like an exercise wellness class, they talk about lactate, how it makes your muscles sore after a workout, because this is primarily how muscle tissue and everything obtains its ATP. Now, in the process of doing this, running pyruvate into lactate, which, by the way, we'll draw lactate on the next slide. That's all I'm going to say for now, actually. We're going to look at this process a little more in just a minute. Okay, so we get lactate. Now, under aerobic conditions, lactate can be oxidized to pyruvate, and you can jump back in the main pathway. Okay. Okay, what about this green arrow right here and the blue one? So this blue one that's going up like this. Pyruvate can be recycled back to glucose 6-phosphate, which is like all headed back towards glucose. So there may be times our body needs to synthesize glucose. What process does that for us? It's a really complex word.
Gluconeogenesis.
Boom. You better not have that figure pulled up. Gluconeogenesis. Got it. Okay, so that is like the opposite of glycolysis. Okay. And pyruvate is the main starting material for that. All right, we take pyruvate. Now there's other things that can feed into this, such as amino acids. We looked at how amino acids, we mentioned in chapter 25, how amino acids are, some of them are what we call glucogenic, some are what we call ketogenic. So glucogenic amino acids ultimately make oxaloacetate, and oxaloacetate is what feeds directly into gluconeogenesis. Okay, so these can be converted to oxaloacetate, which is like the first intermediate. It's like the first product in the pathway of gluconeogenesis. Pyruvate's turned into oxaloacetate. So because amino acids can make oxaloacetate, they can feed into gluconeogenesis. Okay, glycerol though. From lipids, right, triacylglycerols, glycerol can feed into this pathway as well. So amino acids and glycerol feed into gluconeogenesis. Okay. So again, this one's a little more detailed than the one from the figure of your book, but that's on purpose. Glycerol, I do want to add one more thing. So what the way glycerol feeds into this, what just happened? It went somewhere. Here it is. Sorry. Glycerol makes DHAP. That's what glycerol makes. And that is what feeds into. Does anybody remember what DHAP stands for? It's actually part of glycolysis as well. It's made in step 4, and then it's immediately isomerized into glyceraldehyde 3-phosphate. So DHAP stands for dihydroxyacetone phosphate. Glycerol can be made into that and feed right into either glycolysis or gluconeogenesis, depending on what the body needs. Okay. All right, acetyl-CoA. So there's 3 molecules that are just so pivotal in the body, and when we're talking about this main pathway of metabolism. So glucose 6-phosphate, pyruvate, and acetaldehyde. Or sorry, acetyl-CoA. Glucose 6-phosphate, pyruvate, and acetyl-CoA. And we've talked about acetyl-CoA quite a bit. It feeds right into the citric acid cycle, but it is also the end product of beta oxidation. Okay, so the breakdown of lipids. And when we say lipid metabolism, there's a few steps here. Before we get directly to beta oxidation, which actually I should probably write this underneath the arrow because there's lipogenesis, whoops, as well. Depending on, so beta oxidation would be the breakdown. You get acetyl-CoA. Lipogenesis, you can take acetyl-CoA and you can make lipids. But before we can just run beta oxidation, does anybody remember what steps? There's 2 steps that are required.
I might be wrong, but is it like it needs to be moved from like the cytosol into the
That's
mitochondria?
actually, yeah, that's step number 2. There's a transport step. So we need to transport to mitochondria matrix. Before that, we have what we call an activation step, and it requires ATP. We have to make a fatty acyl CoA. Basically, we take a fatty acid, we take the CoA enzyme, and we just got to link them together. And that requires 2 ATP. Invest 2 ATP, which is why when we do our energy calculations on the back end, let's say we're looking at how much energy we get from the breakdown of a fatty acid, we have to subtract off 2 ATP because we invested 2 in the very first step. Okay, once we do those 2 things, we can run beta oxidation. And we get acetyl-CoA, a lot of acetyl-CoA from that. All right, and then, so this was all chapter 24. On the other side, we have our chapter 25 stuff. And this was amino acid catabolism. Okay. So before we actually break down amino acids, we digest them, and then we run several steps. Anybody remember the steps from yesterday? After we digest the proteins and we get them into amino acids or small peptides, there's a few reactions we run. We use transaminase enzymes. So we have step 1 of that is transamination. Okay, where we switch an amino group with a carboxyl group from an alpha keto acid. 2 was, anybody remember the next step?
I know it's like oxidative.
Uh-huh.
I don't remember the last part of it.
Amino clear oxidative. D.
Is it oxidative dephosphorylation? Or am I off?
That's a great guess because we definitely use that word a lot, but deamination. So it's kind of like, honestly, feels a little repetitive from transamination. It's like the opposite though, where we end up with like a glutamate, and then we run it to the urea cycle.
do all amino acids go to the recycle, or do they branch off
No.
and do other things?
So transamination, once we go to oxidative deamination, yes, they go to the recycle. But after transamination, they could go to the biosynthesis of new N-containing, nitrogen-containing. Write small, everyone. Write small, Robin. Okay. So from transamination, they could be synthesized into new nitrogen-containing compounds. But once you do the oxidative deamination step, they do go through the urea cycle, and then they're excreted from the body, right? Okay, but amino acid catabolism can feed into acetyl-CoA and the citric acid cycle. How? This is mainly, so what we've been talking about over here is mainly to deal with the nitrogen, the amino group from amino acids. But the carbons can also be recycled. Okay, so it's the recycling of carbons that gets us to acetyl-CoA. Okay. We really didn't talk through the details of the reverse process, so I will have nothing about the reverse process, acetyl-CoA to amino acids, on the exam. And again, this is where you differentiate between your ketogenic and your glucogenic amino acids, but some are both. So, okay. Look at that beautiful figure. That could have just been your whole exam 4 right there. Okay. Any questions on all the things we wrote?
Can you make it our whole exam 4?
Yeah, can we take a vote?
No, because I've already started writing the exam. But I'm not done writing it, so. All right, look right here. So write an equation, because all you would do is take a screenshot of that, shrink it down, and put it on your note card.
But if that was exam 4 and like it was just filling that out, if you told us not to write a note card, I wouldn't complain.
I agree with Claire. That's a good idea.
Because then you'll only have to study one slide all weekend. But think about how glorious, not this weekend. This weekend's not going to be glorious, but think about how glorious the next weekend is going to be.
Only if we do good on our final.
I'm gonna do my whole extra credit on that picture.
Oh man, that'll make me laugh. Okay, a little bit more on glycolysis. So write an equation for the overall net yield of glycolysis. Which steps are critical energy investing or generating steps, which are important for the regulation of glycolysis? Okay, so I want you guys to try to do this ahead of me before I just write it. What is the net yield of glycolysis? What do we put in? What do we get out? You better write down glucose. Obviously, you got to put glucose in. And it's just one glucose, right? We also have to add, what do we add to glucose? Okay, we don't add this directly to glucose, but what are net wise, what are the 2 other things we put in? Or I should say 2 of something.
ATP?
Yep, that's right, Maggie. 2 ATP. And then what we get out, there's 3 products. I'll give you that hint. Can anybody tell me one of those products? Two of something.
2-pyruvate.
Mm-hmm. Plus, what else do we get?
Isn't it 2 ATP as well?
Yep. And there's one more thing. Two. Maggie, it's on the tip of your tongue. Who knows it?
It's not another ATP, is it?
No, not another ATP. Sorry. Okay, if I'm being very technical here, if we're acknowledging the 2 that we invest, we should say 4 right here. You want to know why? Because net, we get a net of just producing 2. Okay, if we net 2 ATP, if we write 2 on both sides, it looks like we net no ATP. I'm sorry, I should have. Yeah, this has got to be 4 right here. Because we get 2 ATP per glyceraldehyde 3-phosphate that runs through those energy generating steps. So we get 4 total, and we net 2. But we get 2 NADH. Okay. 2 coenzymes, reduced coenzymes. All right. For the exam, also be able to calculate. Remember how to calculate how many ATP we get from glycolysis. Well, we might actually look at that example after we finish this question. Okay, so that's the first part. Now it says which steps are critical energy investing, which ones are energy generating. Anybody remember so there's 10 steps total. Anybody remember which steps require ATP and which steps give us ATP?
Don't. Oh.
I think 1 and 3 give
Mm-hmm.
ATP, but I can't remember which other ones do.
I think, oh gosh, it's 7 and something.
Yes, it is 7 and
Is it 7 and I want to say 10, but I don't think that's it. No.
it's 10. You're right.
It's 10. Okay.
7 and 10. Okay. Now, which steps are important for the regulation of glycolysis? So these have to do with those first 5 steps. There were 2 enzymes that I mentioned that are important for the regulation of glycolysis. And why do we want to heavily regulate this? Because we don't want to waste energy and resources in the body. We don't want to go through making all this pyruvate. And I mean, it's 10 steps. It's a lot of work for the body to do. It's a lot of enzymes. So we don't need to do all this if the body simply doesn't need energy. So there's 2 regulation points within the first 5 steps for that reason.
Was step 4 one of them?
Step 3 was the
Oh.
biggest one. So the energy investing steps are actually the ones we regulate. So in step 1, we get glucose. And what we get out is G6P, glucose 6-phosphate. Anybody remember which enzyme runs this reaction for us?
Is it hexokinase?
Uh-huh. So we got hexokinase, and then we invest in ATP here. And we get an ADP. Okay. So G6P can come back, and sometimes we'll put an arrow or like a kind of a line like this to show that it inhibits. And it's a competitive, competitive inhibitor. If you read through chapter 22, it also tells you it can be an allosteric inhibitor. So if it's competitive, it can bind the active site. Okay. But let's say glucose is already in the active site. It can bind another spot. Well, this actually wouldn't make sense. If glucose was already in it, then it cannot. Allosteric inhibition, it binds somewhere different from the active site, and then it causes a conformational change so that the active site is no longer available for the substrate to bind, for glucose to bind. So it could bind a location different from the active site. Okay. And it can shut off hexokinase that way. Or, yeah. Okay, step 3. Is a little more complex as far as the enzyme that we get. So the reactant on step 3 is fructose 6-phosphate. And we're going to phosphorylate it, and we get, I'm going to abbreviate, fructose 1,6-bis, and then I put a P for phosphate, fructose 1,6-bisphosphate.
Yeah, I have a question.
Yes, Maggie.
Where did this biz come from?
It's just the name. So
BIS?
BIS, bisphosphate. Why they don't call it diphosphate, I don't know. So they just, I mean, di usually indicates 2, right? Like 2 OHs, dihydroxy, or so we've used di as a prefix for 2. So it would make a lot of sense to me if it was fructose 1,6-diphosphate. But that's not the name of it. It's fructose 1,6-bisphosphate. So
Okay.
that's one of those things. I wish I had a good explanation for you, and I don't. But the enzyme here is the major piece to focus on. What's the enzyme? Anybody remember? It's another kinase.
Kinase.
Yeah, it's a kinase because we're phosphorylating. So it's phosphofructokinase. All right. So there's several regulators here. We have some activators and we have some inhibitors. So activators, I'm going to write in green because green means go. So ADP, well, I should start with AMP. AMP and ADP, okay? So these tell phosphofructokinase to run this reaction. And in black, the ones that inhibit ATP and citrate. So if energy is in good supply and we have a lot of ATP, we inhibit this because we don't need to run it. We don't need to make more ATP. If ATP is low, then the concentrations of ADP and AMP are high in the body, and those act as positive allosteric regulators. Positive allosteric regulators for phosphofructokinase. Okay, any questions on that? Next thing, so again, from glucose, I want you to know the names of the intermediates for the first 5 steps. Know these enzymes and the kind of regulation that happens for these. Okay, describe the mechanism for the regulation of glucose metabolism. So that sounds redundant. You're probably like, we just did that. I will differentiate on the exam. If I say the regulation or the inhibition stuff for glycolysis, that's what we just talked through. But if I say glucose metabolism regulation for glucose metabolism, I mean something different. I mean, when we have conditions of hypo and hyperglycemia, we have hormones that respond to help regulate our blood glucose levels. This is what I mean by glucose, regulation of glucose metabolism. Anybody remember which two hormones do this?
Insulin and glucagon.
Insulin and glucagon. So this is what I'm talking about with glucose metabolism, insulin and glucagon. I want to know how these work and when they're stimulated. So we have insulin. Written in red. Okay. Now, when does what conditions, I should say, under what conditions is an alarm going to go off in our body that we need to synthesize or release active insulin?
When your blood glucose is too high.
When our blood glucose is too high. That's exactly right. So I'm going to use the chemistry word hyperglycemia. So that means glucagon is the opposite. When our blood glucose is too low, hypoglycemia. That is the condition that would stimulate the release of glucagon. So in a hyperglycemic condition, what is insulin going to do? What are some specifics? How is insulin going to help? So we have hyperglycemia, we have too much glucose in our blood. We've got to bring it down. So what are some things that insulin can help do to bring blood glucose concentration down?
I think one of
gonna-
them was like, oh, sorry, go ahead, Robin.
No, you already answered.
I was just gonna say one of them, insulin increases the cellular blood. I can't remember the last word.
The uptake of glucose. Yeah, so cells are going to start to absorb more glucose. So one thing, it will increase I'm going to do a capital G for glucose. Anybody remember another thing it will do?
It's gonna synthesize your glycogen in your liver.
So we're going to run glycogenesis. So we're going to synthesize So we're going to take glucose out of the blood and we're going to store it as glycogen. So synthesize meant to write S-I-Z-E, synthesize glycogen via glycogenesis. Anybody remember another thing it will do?
Does it synthesize lipids and proteins?
Yes, because I'm going to write that down on number 4 because okay, because we're going to really ramp up glycolysis. So we're going to take glucose, we're going to metabolize it through glycolysis, break it down into pyruvate. Pyruvate will be made into acetyl-CoA, and we got all this acetyl-CoA, and we can run lipogenesis, make proteins, and that's one way we can use up our glucose. Okay, so we can ramp up with glycolysis. You could say metabolize glucose via glycolysis. Okay, that's insulin. And remember that insulin is stored as a zymogen. It's one of those proteins that we just want to sleep because in a moment's notice we may need it. And then we snip some sort of covalent modification, and we get the active insulin enzyme. Okay, what if we have hypoglycemia? What if we have too low of levels of glucose in our blood? Our glucose concentration is low. This is where glucagon is going to be signaled.
And slow down the cellular uptake of glucose. Oh, by the way, on the quiz, you put like update or something on one of them instead of uptake.
Oh, really?
Yeah, it just kind of made me laugh. I just want to let you know.
I realize there's definitely typos on those quizzes. I try to go back and read through them, but
Well, I just stared at it for a second because I was like, okay, it doesn't say uptake. Is she trying to trick us? But then I'm like,
no,
she doesn't.
no, that's an honest mistake. I really try not to trick with typos. So it's going to slow cellular uptake. It's going to metabolize or break down glycogen. So this is genolysis. We're going to run gluconeogenesis. We're going to take protein and lipid metabolites, and we're going to filter them back through gluconeogenesis to make more glucose, and we can dump all that into our blood. To run, so essentially we'll also, as well as running gluconeogenesis, we'll break down lipids and proteins. Okay, so I had a picture for this on the slides that shows you that the pancreas up here, and these are going to release your insulin or your glucagon, and that's going to go to your liver. Hold your applause, please. At my drawing, it's going to go to your liver, where we have glycogen. Okay, so glycogen is either going to be stored here. We're going to bring glucose in and store it, or we're going to take glycogen, break it down into glucose, and dump this into our bloodstream. Okay, so that is glucagon. I guess I could write it a different color. Insulin does the opposite. I'll do insulin in blue. So insulin is going to pull glucose out, and then it's going to store it as glycogen. So I'll do these little black dots for our glucose. So insulin pulls it out, glucagon dumps it in. Okay, that's a regulation of glucose metabolism. Now, what is one major result or consequence of a faulty system here? Like if we have a breakdown in these hormones, particularly insulin, what is the condition that can arise from that?
Diabetes.
Diabetes. And this is, we have an issue with our insulin. Maybe our body doesn't produce enough of it or doesn't produce it in the active form. We have to inject insulin at that point into the body regularly or not necessarily regularly, but at certain times when we need it. Okay, like after a meal or something.
If that's on the test, nobody better miss that. Just think about it. Just look at me.
Wait, say it one more time then.
If we have a breakdown of our insulin function, the result of that is type 1 diabetes. Okay, so that's a condition that is the result of a faulty system here, some kind of flaw with insulin. There's many different things that can cause that. And there's a lot of mystery behind sometimes why diabetes is kind of arises in people, particularly youth. And so, anyways, I'm not going to drill you on diabetes, but I might ask you, describe a situation that diabetes is the result of or something. Okay. What time is it, by the way? 9:53. We've been going for a little bit, and I still got 1, 2, 3, 4, 5. I knew I would have too much. We're kind of walking through this slowly. I may pick up the pace here for these next couple slides. I want to get you through a few of these things. Pyruvate. Pyruvate is one of the molecules that I said was super versatile, okay, pivotal. It can go different directions in metabolism. So under aerobic conditions, we're going to oxidize, or we say aerobic oxidation. We're going to make acetyl-CoA, which is just CH3. We're just going to link up this acetyl group right here with coenzyme A, and we get CO2 with that. Okay. Now, if we're going to oxidize pyruvate, something's got to be reduced. So we need NAD+, and we reduce it to NADH in this step. Okay. So that's a key thing to know, is that we reduce the coenzyme NAD+ in this step. Okay. Make sure you're writing this stuff down with us. In anaerobic conditions, this is when we have little to no oxygen supply. We're going to reduce pyruvate to lactate. Okay, so what's going to happen is we're going to add a hydrogen on right there, and we're going to add a hydrogen on to that carbon right there. We're going to reduce it. So when that is done, what this looks like is CHOH, C-double-bond-O. Okay, this is lactate. And guess where those hydrogens come from? It's the opposite of the oxidation above. We take NADH and we remake NAD+. Now, why do we care that NAD+ is regenerated in this step? This is kind of the key important thing here. I don't know who's calling me. The key important thing here. Decline. Sorry, everybody.
You never know who's calling you.
I never know. It's always a random number. It could be the ENT doctor. I've got to take my son in next week to get his ears looked at because he's got to get tubes. And so I'm wondering if it's their office, but I don't know why they would be calling me. I know that I need to fill out some paperwork. Okay. Or it could be spam. Do y'all still get those? Your auto warranty is blah, blah, blah. I hate those calls. My husband is really mean sometimes. He told me what he did too. Sometimes when random telemarketers call him, he like plays along with it. And I'm like, one, I don't have the patience or the time to do that. But he like really ticks him off. It's not nice. Okay. So we regenerate NAD+. Why do we care that NAD+ is regenerated?
Is it so that lactate can be converted back to pyruvate to be formed into acetyl-CoA later?
No. No, it's not that. So remember, these conditions means we're in like red blood cells or an organism that doesn't have oxygen or mitochondria. And so somehow we have to we still have to supply ATP to those cells and those tissues. So what happens is that the way we get ATP is simply from glycolysis. Now, how many ATP does glycolysis produce? 4 total, net yield of 2, right? But that's what happens. So we take lactate. This NAD+ is going to go help continuously run glycolysis so that we can produce ATP for those cells in those tissues. We have to continuously supply NAD+. And this process right here supplies the NAD+ to just continuously, it's a closed loop, run glycolysis to produce ATP for these tissues and these cells that do not have the means to make it via oxidative phosphorylation. That is why we care that NAD+ is regenerated.
Will you write that down?
So NAD plus, is that a catalyze, it like catalyzes the reaction?
It is a coenzyme in the reaction. It's not the actual enzyme. It's a coenzyme. We actually didn't even talk about the enzyme here, but it's just a product. It's technically a product of the reaction, and it just goes right back. Does anybody remember what step in glycolysis NAD+ is used or is used for? It's step 6, right between the production of glyceraldehyde 3-phosphate. We take glyceraldehyde 3-phosphate and we oxidize it. And in the process, we need NAD+ to do that. That's NAD+ is the oxidizing agent for step 6. And then in step 7 is where we produce ATP in glycolysis. Okay. So wait, Robin, what did you want me to write down?
I got it. You just said it.
Okay.
I just have to, if you put this up, I'll just have to go back and write everything.
The particular figure I'm thinking of that shows you all this is right here. Right here. Okay. So we take pyruvate, we reduce it to lactate, and in the process, we remake NAD+. NAD+ goes back and feeds step 6 in glycolysis so that we can make ATP. So it's just this closed loop that keeps going and keeps going. Okay. But that's the whole purpose.
Are you gonna upload this too?
You want me to?
Yeah.
The recording?
Yeah, because like I don't want to make you like I want to get through it so we can
Yeah.
get through all of it.
Yeah, sure.
Okay.
All right. So that's pyruvate. I definitely want you to know anaerobic and aerobic conditions, like how pyruvate serves both of those things for the body. Okay. Body has 2 main energy producing pathways. Under what conditions is each one used? Okay, so this is kind of a big picture question. The body has 2 main energy producing pathways. The first one obviously is glycolysis. But what is the second one? What other pathway produces a lot of energy? And I don't mean oxidative phosphorylation. Glycolysis feeds directly into electron transport and oxidative phosphorylation. What is the other one that we can yield a lot, even more than glycolysis, a ton of ATP from? Abby, I think you said it.
Was it beta oxidation?
Yes. Okay. So I want to be simple here. When glucose is in good supply, or when we have glycogen storage, we'll run glycolysis. Why? Because it's quick, it's easy. There's few steps. The breakdown of the storage molecule is simple. We still get a decent yield of ATP from it. So when okay, our diets typically tend to contain a lot of carbs and a good supply, continuous supply of glucose. So our body just uses that. When is beta oxidation used? When glucose and glycogen supplies or stores have been depleted. That's when we run beta oxidation. All right, pretty simple there. Don't overthink that one. Okay, what kind of biological molecule is used in beta oxidation? What kind of molecule are we obtaining this energy from? What are we breaking down and metabolizing in beta oxidation? So for glycolysis, we use a carbohydrate. We use glucose. Now, I didn't mention this, but other monosaccharides, fructose, galactose, other monosaccharides can feed into glycolysis. We have to manipulate them a little bit first, but they can also be fuels for glycolysis. But the main source is carbohydrates. What's the main source for beta oxidation?
Isn't it fats?
Lipids, specifically triacylglycerols. Okay, we rip off the fatty acids and we break down all the fatty acids and get all the ATP from it. Okay. Questions on that? Okay, 3 slides left. Here we go. What is the difference between glycogenesis and glycogenolysis? What enzymes are involved in these processes? So the difference is the synthesis of glycogen or the metabolism of glycogen. So glycogenesis is the synthesis of glycogen. Glycogenolysis is the metabolism. Glycogen. All right, so the way this works, if we take glucose 6-phosphate, this is our guy. The first thing we're going to do is we're going to convert it to glucose 1-phosphate. Anybody remember the enzyme that does this? I don't expect it, honestly, at this point. It's a mutase. Phosphoglucose mutase. It's a very long word. If I can shrink that down. Okay, phosphoglucomutase, that's the enzyme. Then once we have G1P, we take it to the liver where we store glycogen, and basically the next important enzyme is glycogen synthase. And remember that UTP, UDP molecule I was talking about? It's kind of the chauffeur of glucose 1-phosphate, chauffeurs it to the liver, and then we add it to glycogen. We dephosphorylate it, take the phosphate group off, and then we just link these monosaccharides of glucose together. They are all linked together. So if we have, if we have one glucose here, this is linked to the next one via a, anybody remember what kind of linkage that is?
Alpha, 1,4 alpha linkage.
Alpha 1,4. Those are the kinds of glycosidic bonds that hold glycogen together. All right, so the only thing that's different is when we go to break down glycogen, we use glycogen phosphorylase. And glycogen phosphorylase is going to break the glycosidic bond and I can't even speak right now. Add a phosphate group, phosphorylate glucose simultaneously. So it'll break the glycosidic bond, phosphorylate, we get G16P. We run it back to G6P, and then we can feed it through glycolysis. Okay. Do we need like a little break? Are y'all getting kind of tired? We're getting close to being done. I want to finish this for y'all. What do we got? We got gluconeogenesis. We'll do this question real quick, and then we'll take like a 5-minute break if anybody needs to go to the bathroom or get a drink or something, and then we can finish up. This last one is just the practicing calculating how much energy you get from fatty acids. Okay, so that one's going to be some math. This is really only the, what 2 steps required for, you know, we mentioned these earlier, these first 2 steps, but we'll do that. So real quick, let's name 3 molecules that can feed into gluconeogenesis, and then we'll take a 5-minute break. I might go grab some water, and then we'll finish up. Okay, gluconeogenesis. Someone real quick, define it for me.
It's the creation of glucose.
Creation of glucose.
Right?
Isn't it also through like things that aren't like strictly carbs?
Yeah, a lot of different things can feed into gluconeogenesis. Amino acids. Pyruvate is the main thing. Okay, so the main reactant is pyruvate. Okay, so if we're naming 3 molecules that feed into it, pyruvate would be the first one. Can anybody name 2 others? Abby, that's kind of what you were getting at.
I was gonna say like proteins and lipids.
Yes, but I want to be a little more specific.
Is it amino acids and triglycerols? No, am I looking too far?
Amino acids and glycerol, yes. I want to be even more specific than that.
Okay.
So I'm going to scroll down to the figure. I think it's the very last slide here. I want y'all to look at this figure. Here it is. So we said pyruvate right here. Pyruvate is the first thing, but we see there's some other entry points.
That pyruvate carboxylase and that phosphono-pyruvate carboxykinase.
Where are you even reading?
Oh, I don't know. My brain's not working.
I think you're reading these enzymes. These are the enzymes that run the stuff. So glycerol is one thing.
Oh, glycerol and glucose.
No, glucose is the
Oh, dang. Just ignore me. Hey, I'm making you laugh. Bonus points.
Glucose is the final product.
Ah.
Oxaloacetate. Remember why we called some amino acids ketogenic and glucogenic? Because we said they ultimately make oxaloacetate, and that can feed from the citric acid cycle. That's the last product of the citric acid cycle. And that can feed directly into oxaloacetate or into gluconeogenesis. Okay, so glycerol, you could write DHAP if you wanted, oxaloacetate. I would take all those answers.
Do it needs to be all 4 or just 3?
I just said name 3, but I was giving you 4 examples.
Okay.
Okay. All right, let's take a break, and then we're going to focus on, so that was a lot. This was mainly all just chapter 22, a little bit of chapter 24. Then we're going to do a little bit of chapter 24 down here. Some of the big hitters from chapter 24.
This is a lot of the stuff we need to focus on for the exam, right?
Why are you saying it in that tone of voice?
Just answer the question.
Of course it is. Of course. Okay. Why would I be reviewing it if it wasn't?
I don't know. I just feel sick. Like,
do you need to inject some insulin?
I don't know, like literally and figuratively, I'm like sweating and I do not feel well.
You need to go take a nap? Did you stay up too late doing physics homework?
No, I didn't even do physics yesterday. I said, screw physics. I'm going to sleep.
Uh-oh.
Yeah, now I feel like I'm behind, but I'm not. It's okay.
Okay. All right. Well, take a few minutes. I'm gonna go. Actually, I have a decent supply of water. I'm gonna see if they left me a voicemail. Nope. Jonesboro, Arkansas. I just don't recognize this number. I'm tempted to call them back. Do y'all take calls from random numbers?
No, I always call them back if they don't leave a voicemail. I'm like, who is this? Oh, talking about that freaking, how you said the car people called about the warranty, whatever. I just remembered, I thought I was awake, but I was like half asleep this morning. And they called me at like 8 o'clock.
Called you at 8 a.m.?
Yeah, and I was like, who am I talking to? It was like one of those where you're like still half asleep, but you're awake, and then you don't remember if it was a dream or if you were
Okay,
actually
I
awake.
have a crazy story on this. So my best friend, one of my best friends from like middle school, she lives in Nashville now, Nashville, Tennessee. And she was telling me that her husband like stays up late and plays video games. So he was still awake, but she was asleep, and she says she could remember she was having this like crazy, crazy dream that like literally she was somewhere in her hometown, and like all of a sudden like a bunch of people she knew from school were there, and they were being ambushed by somebody, like open fire, like gunshots, all this stuff. So it's like a really stressful dream. And she said in her dream, she's like frantically trying to find her phone and call 911 because they're being shot at. And her husband told her the next morning that like a police officer had come to their house at like 2 a.m. and knocked on the door and was like, "We got a call. Is everything okay here?" And she was like, "What?" So the only consensus possible is that while she's having this crazy dream, she actually calls 911 on her phone. I just wish I had been like the like 911 responder
Operator.
on the phone who was like talking to this asleep girl at 2 a.m.
Oh my gosh, that's funny. I've woken up and seen that I've called 911 before.
Really?
Yeah,
Like
but
in
it
your
wasn't.
dream?
No, it wasn't because of dream. It was because of under the influence.
Oh no,
I don't
Robin.
know why I called. Oh my gosh, one time I was on the lake. This was last 4th of July. I was on the lake. And so like my parents are really like, if I call and don't answer, they always think I'm like dead on the side of the road or something. Or they call and I don't answer, something weird. So like my phone, I was on the boat and like it, just a bunch of people tied up. We were just partying on the 4th of July. And I don't know what happened, but my phone, you know how the iPhones have that feature where you can like triple click and it like sends like an alert and it'll call 911.
No, I didn't know you could do that.
Well, mine has it. I can like triple click or something and it'll like send a text to my mom, my dad, all my emergency contacts. So like my mom, my dad, my sister, my best friend, and be like, Brianna's at this or Robin's at this location, like whatever, this like she's in trouble, blah, blah, blah. And my phone did it twice. And it called 911 twice. And I was like, and then I looked at my phone like on the boat and I was like, what the heck? And my mom and dad were like blowing my phone up. They're like, are you okay? Are you okay? And I was like, yeah, I'm just on the boat in the middle of the lake. Like, so I'm just like thinking what the 911 operators heard. Probably just a bunch of kids partying. They're like, oh, they're fine.
Wow.
Yeah, that was
That would freak me out.
Yeah, I was like, what the heck?
That would freak me out. Okay. My iPad is not reading my finger right now. All right, well, we will, I mean, y'all can still see my screen, right? It's like not. Come on. Hold on. Let me try to open this back up. No. Maybe it will now. Okay. So let's finish these last two slides. What two steps are required for a fatty acid to enter beta oxidation? And then let's name the 4 steps of beta oxidation. So we actually wrote these first 2 steps, if y'all remember, on like the first slide. So number 1 is called activation. And we have to join a fatty acyl, or sorry, we have to join a fatty acid to acetyl-CoA, and in the process we produce a fatty acyl-CoA. Could y'all draw that? If I ask you to draw that? Acyl is what? It's a carbonyl, some kind of R group. This is the long hydrocarbon chain of the fatty acid, and it is connected to acetyl-CoA through a sulfur. This requires 2 ATP. Step number 2. Oh, I'm really leaving myself a lot of room here, aren't I? Let me shrink this down. Step number 2 is transport to the mitochondria matrix. All right. Beta oxidation. Someone can tell me the 4 steps. I know they can. I think I'm good. Okay. Who can tell me the 4 steps? The first one is, this one should be the easiest. A beta oxidation step. Okay, the first beta oxidation. So step number 1 is actually a beta oxidation. We're using a dehydrogenase enzyme, and we are taking the beta carbon. So this right here, I'll change colors, I guess, would be the beta carbon. We're going to oxidize it. Or sorry, we're going to oxidize it by removing hydrogen. So oxidation is the addition of a carbon-oxygen bond or the removal of hydrogen. So this has 2 hydrogens. The one in black is the one we're going to take off. This is the alpha. So these 2 hydrogens in black, we're going to remove those. And then we have carbon double bonded to carbon. This is the beta. This is the alpha carbon. And there's the okay, it's a dehydrogenase. So step number 2 is a hydration step. And we're going to add water. We're going to turn the beta carbon into an alcohol functional group. So I might actually just continue. So this would be step 1. I'm just going to continue step 2. We're going to, well, hold on. Let me finish right now then. So now this is going to be OH. This one's going to have the H. We broke the double bond again. This, by the way, will still have 1 hydrogen each attached to it. Step number 3 is the second beta oxidation. And then step number 4, we cleave the bond. So step number 3 is beta oxidation again. Step number 4 is we cleave the carbon-carbon bond between the beta carbon and the alpha carbon. So let's draw this real quick. So we're going to oxidize this. The beta carbon is now going to become alpha and carbonyl carbon. Now going to become double bond to oxygen. And then this will end up with 2 hydrogens back on it. That's the alpha carbon. And then in step 4, we're going to cleave that bond. Now there's 2, this is not part of the question, but this is important for the exam. There are 2 coenzymes involved, step 1 and step 3. So in step 1 here, we take FAD and we reduce it to FADH2. This is step number 2, the hydration step. So we have water coming in. Step number 3, we take NAD+, we reduce it to NADH. And this is important for the energy calculation later because we know FAD will ultimately generate 2 ATP and NADH will ultimately generate 2.5 ATP. Okay. I didn't talk about the enzymes a whole lot, but please know what kind of enzymes. Dehydrogenases, hydratase, thiolase. And that is straight off of a slide of chapter 25 or 24 if you need it. All right, last problem, and then we will definitely have been well past 45 minutes to an hour, coming up on an hour and a half. How much energy is released from ATP from the complete oxidation of lauric acid and then palmitic acid? If we do one, you should be able to do the other. Let's just look at palmitic acid. Okay. So it's the most common fatty acid found in humans, plants, all microorganisms. How much energy is released as ATP from the complete oxidation of palmitic acid? So anybody can help direct us here. What are the steps that we need to do?
Don't you need to first figure out how many carbons are in palmitic acid and then you divide it by 2?
Yes, that's exactly right. So how many carbons do we have? Whoops.
1, 2, 3, 4.
All right. So the first step is we need to figure out how many acetyl-CoA are we going to produce from this. So we do the number of carbons divided by 2 should give us how many acetyl-CoA?
We get 8, right?
Yep. Then what do we need to do to 8 acetyl-CoA? How many ATP are we going to get just from the production of 8 acetyl-CoA? For every 1 acetyl-CoA, how many ATP do we get?
Isn't it 10?
0.5.
Oh,
I actually messed that up on exam 2. But 10.5 to be technical and accurate. Okay, so 8 times 10.5 is 84. All right, what's the next thing we need to do? Well, remember that every time we run a cycle of beta oxidation, we get how many ATP just from the coenzymes produced? Because these feed right into electron transport, oxidative phosphorylation. Each cycle of beta oxidation, 2 plus 2.5, is going to give us 4.5 ATP. So we need to ask ourselves, how many cycles of beta oxidation is going to take to totally break this thing down?
Okay, I noticed this. I don't know if it's like this for everything, like all of them, but when I figure out how many acetyl-CoA, it's usually one less. The beta oxidation cycle is always usually like one less. Is that right for everything?
Exactly right. Here's
Okay.
why. Because the very last step, you produce 2 acetyl-CoA. So it takes one less cycle. So we're going to cleave right here. We're going to cleave right there. That's 2 cycles of beta oxidation. Each of these produces an acetyl-CoA. Acetyl-CoA. We're going to keep going. Every 2 carbons we cleave because we shorten it by 2 carbons every time. Okay. So on this last run of beta oxidation, we're going to get acetyl-CoA and acetyl-CoA just from one run of beta oxidation. So it'll be 7. If you count the number of little lightning bolts I drew, 1, 2, 3, 4, 5, 6, 7. Yes, it's always one less than the number of acetyl-CoA you produce. So we run 7 cycles of beta oxidation. And we know for every 1 cycle, we get 4.5 ATP. So how many ATPs, how much ATP is produced just from beta oxidation?
This may be wrong. I'm bad at math. 30.5? 7 times 4, 7, 14, 21, 28. 0.5 times 2 is 29. No, that's wrong.
No, it should be 31.5.
31.5, yeah.
Half of 7 is 3.5. So you add
I'm
that to 28. You're good. 31.
Calculator for sure.
So far we got 84 ATP plus 31.5 ATP minus 2 ATP because we had to invest those in the activation step. Total, that's how much? 84 plus 31.5 minus 2.
$29.5.
How many, Maggie?
29.5.
Did you do 84 plus 31.5?
Oh no! Wait a minute. No, I subtracted
84
2 from.
plus 31.5 minus 2.
Isn't it 113.5?
That's what I got. Okay, everybody. Was that a decent review? And again, I didn't even get into chapter 25, but we did go over it yesterday, so it should be the most fresh, I guess. And it was the fewest number of slides. From chapter 25, I'll just write down a couple things. Know the process of amino acid catabolism. Okay, so that includes trans, amination, oxidative deamination, know the urea cycle, which is technically like the third part. So I should have just said another little dash. Okay. For sure, know the essential, no, hold on. Ketogenic versus glucogenic amino acids. So remember I said you didn't have to know all of them, but at least know 2 examples of each. And then also know the essential amino acids. And so know 2 of those, and know the purpose of them, and then the deficiencies associated with them in the body.